When we begin going through this passage, we notice that there's a table at the bottom with results. And what this means is that immediately our train of thought should be thinking, all right, experimental type of passage within the biochem section. We should be looking for a reason for the study, potential control and experimental groups, independent and dependent variables, as well as any conclusion or results that they obtained from the study. As we begin, we see that they're talking about blood flow, and anything about blood flow, cardiovascular system should immediately be going through our mind. Then they relate it to the MR process that we're not too familiar with, but let's continue. We see that they start talking about a chain of events, and you'll notice this in a lot of biology or biochemistry passages where they say A affects or activates B, which activates C, which activates D. And you kind of, maybe if you have time, want to write it down or just mentally keep track of it. But I wrote down a cascade of events that I saw happen. So we have the cell membrane depolarizes, then VSM contracts, then the voltage-gated calcium channels open, the systolic protein in the cell calmodulin is bound to, which then activates the enzyme that phosphorylates LC20, which then activates the myosin head, which binds to actin, which forms the cross bridge, and ultimately causes muscle contraction in skeletal muscle. So this is a chain of events that one thing activates the next thing. And if one thing is disrupted, the whole entire cascade might be disrupted or is most likely disrupted. As we continue going through, we see that they're talking about actin polymerization and how when actin is polymerized, it turns the G actin into F actin. And it says that it can be blocked with the drug. So what this means is that as more and more actin are polymerized, the ratio of F actin is going to increase relative to the G actin. Because the G actin would be decreasing in number, the F actin would be increasing as actin polymerization continued. Then we read through that the goal of the study is to examine changes that occur in VSM during the MR. We find that the independent variable is increasing internal arterial pressure, and the dependent variable, the consequence of this is that arterial diameter is affected, phosphorylated LC20 is affected, phosphorylated calponin, G-actin is also affected, as we see in the table. Then, as we continue looking at the table, when we're analyzing the results, we see at high pressures, we have a very low diameter. So at high blood pressure, there's vasoconstriction. And then at that high pressure, we also see high phosphorylation levels, high calponin levels, but we notice that they're not different than the middle value. So that might be something to keep in mind. And we notice that there's very low G-actin, which, referring back to the passage, we know that if there's low G-actin, that means there's high F-actin. So this is pretty much a summarization of how you would break down this passage. In the beginning, there was information that we kind of knew about, not too much. They talked about this new process or MR that we might not be too familiar with. They showed us a cascade of events that occur. Then they kind of introduced their experiment, and then they showed us the results with the table. Question 1 asks, based on the information in the passage, which type of enzyme is most likely to suppress CBC? And what we do is we look back into the passage, and we notice that CBC is part of that cascade of events that we drew out. So we look back into that cascade and see anywhere where an enzyme would be part of this cascade. And we notice that it says an enzyme that phosphorylates amino acid 19 of the myosin light chain LC20. So there's an enzyme that phosphorylates that amino acid. And the question is asking, what would most likely suppress CBC? So if there was an enzyme that phosphorylated LC20, an enzyme that prohibits or takes away that phosphate would reduce the effects of this domino effect of the cascade and by default lower the CBC activity. So there was an enzyme that phosphorylated LC20, which domino effect and activated CBC. But if we find an enzyme that would remove a phosphate group, we would slow down that domino effect or even inhibit that domino effect, and therefore CBC activity would be diminished. So looking through the answer choices, we want to find an enzyme that would take away a phosphate group. And going through the answer choices, we have phosphorylase, kinase, phosphatase, and synthase. And the correct answer here is phosphatase, because phosphatase takes away a phosphate group from the substrate. And an easy way to remember this is phosphatase. The end part of phosphatase sounds like taste. You're barely just getting a little taste of that phosphate group before it's removed from you. So phosphatase comes and removes or yanks away that phosphate from you forcefully, and you only got a little taste of that phosphate before it was removed. Question 2 states, which statement regarding VSM function and cytoskeletal dynamics is best supported by the data in Table 1? And reading through the answer choices, this is one that we have to read the answer choices before we can eliminate them. There's no way we can come up with an answer beforehand. So we have to go through every single answer choice and then see the one that is best supported by the data. So question A, or option A, reads, vasoconstriction is associated with an increase in the ratio of F-actin to G-actin. And if we go back, this is true. What we notice is that during vasoconstriction, that is when the arterial diameter is the smallest. So if we look in Table 1, the very right column, arterial diameter is 175 micrometers, and that is the smallest relative to the other ones. And we notice that at this level, we also have the lowest G-actin amount. And remember from the passage, as actin is polymerized, it goes from G-actin to F-actin, meaning that if there's low G-actin, there's high F-actin. And what question or option A states is vasoconstriction, so small diameter, is associated with an increase in the ratio of F-actin to G-actin. And that's true because the G-actin is the lowest at that point and the F-actin is the highest. Continuing to answer choice B, they say F-actin levels decrease as phosphorylation of LC20 increases. So the phosphorylation of LC20 was part of that whole entire cascade of events, and the end result of that cascade was vasoconstriction. And this is saying that phosphorylation of LC20, which is part of that cascade, F-actin levels decrease as that cascade continues. And this is actually the opposite. As that cascade continues and as we get vasoconstriction or contraction of the skeletal muscle, F-actin levels increase because G-actin levels decrease. So B is actually the exact opposite of what happens. Answer choice C says vasodilation does not affect phosphorylation levels of calponin. And this is false. Phosphorylation levels of calponin was part of that cascade. Vasodilation would possibly affect it because as we vasodilate, the levels of phosphorylation of calponin actually would decrease. And if we look in the table, the row that says phosphorylated calponin is affected. It goes from 7 to 18 and 18 as the arterial diameter decreases. So we would expect the opposite to happen. We would expect it to increase as vasodilation happen. So when we went from 225 micrometers of the arterial diameter all the way down to 193 and then 175, the phosphorylated calponin levels went from 7 to 18 to 18. During vasodilation, we would expect it to go from a high number down to a low number. Option D says arterial diameter reduction is always dependent upon increased calponin phosphorylation. And this isn't true. It's saying it's always dependent. Arterial diameter reduction is always dependent upon increased calponin phosphorylation. This is not true because if we look in our table, in the row that says phosphorylated calponin, we see that at 193, we have phosphorylated calponin percentage of 18. And then at a arterial diameter of 175 micrometers, we have a phosphorylated calponin level of also 18. So the arterial diameter changed, but the phosphorylated calponin levels did not. Question 3 asks, compared to control RMCA held at an internal pressure of 120, application of latrosulin B to RMCA held at 120 will most likely result in... And for this, we have to go back to the passage where they discussed drug B. And they did so when they were talking about G-actin and F-actin and how actin depolymerization can be induced by the drug. So if we had a control group, that is exactly what we had in the passage. So the control group would be the one at internal pressure of 120, and the experimental group would be the same ones at 120, but just with a drug added. And if we go back and look at the drug, it induced depolymerization, and this would take away polymerized F-actin and convert it back to the G-actin. So drug B would increase G-actin levels and decrease F-actin levels. So that's what we should be thinking going through these answer choices. We're looking for something that talks about G-actin levels increasing or F-actin levels decreasing. And we go through, A talks about phosphorylated LC20 levels. That doesn't really have anything to do with the actin depolymerization. That has to do more with the cascade of events that we were talking about earlier in paragraph 2. B talks about decreased arterial diameter. And that is the same thing as vasoconstriction. Decreased arterial diameter is the same thing as vasoconstriction. And that also is a part of the cascade of events that we noticed in paragraph 2. So paragraph 2, if that whole entire cascade of events occurs, we get muscle contraction, which also results in vasoconstriction. So that A and B are both out. Then we look at C. C is talking about exactly what we were looking for, decreased F-actin levels. Because as the drug depolymerizes the F-actin would convert back to the G-actin, so the F-actin levels would decrease, G-actin levels would increase, and C would be correct. But let's see why D is also incorrect. D states increased phosphorylated calponin levels. And this isn't necessarily true. If we have the drug that inhibits the G-actin levels, we would not see increased phosphorylated calponin levels. We would actually see decreased phosphorylated calponin levels. And this is because if you look at the table where they're talking about G-actin, at an internal pressure of 10, it is 1. At an internal pressure of 80, it is 0.3. At an internal pressure of 120, it is 0.1. And the phosphorylated calponin levels are 7, 18, and 18 respectively. So as we increase the G-actin levels, remember as the F-actin are getting depolymerized, they'll turn back into the G-actin. As we're increasing the G-actin levels, the phosphorylated calponin levels actually decrease, not increase. So D is actually the opposite of what we would see. Question 4 asks, according to the information in the passage, which statement best describes the function of the MR in response to stimulation of the sympathetic nervous system? The MR would, and as soon as we read this, I highlighted the word sympathetic nervous system. So sympathetic nervous system is your fight or flight response. Let's say you're walking through the woods and all of a sudden you see a mountain lion. You're either going to run as fast as you can or fight. I don't know anyone that would fight, but you would run away from the situation. You'd have adrenaline flowing, all the blood in your body, most of it would be going to your muscles to help you run or to your brain to help you think of a way to escape. They would not be going to your stomach or small intestine that is focused on processing food. So fight or flight response, we are getting all the blood to the extremities. And the answer choices read, the MR would A, equalize blood pressure in cerebral resistance arteries and the aorta. And this is incorrect because the aorta is right where blood leaves your heart. And if this was equalized with the arteries in your body, blood would not be flowing from your heart to those arteries. There would be an equal pressure. It wouldn't really be going at any pace to those extremities. This would actually be very bad. That would mean your blood pressure is pretty much non-existent. There's no difference in the blood pressure from your heart to anything else. There needs to be a difference so that the blood continues flowing. Answer choice B reads, the MR would moderate blood flow to the brain under high pressure. And this makes sense because blood would be going to the brain, and the MR was our response in the changing of arterial vessel diameter. So it would make sense that the MR would moderate this blood flow, making sure that the blood to the brain wasn't very high or it wasn't too little. Option choice C says the MR would enable cerebral resistance arteries to locally vasodilate. During sympathetic nervous system arousal, you would not vasodilate. You would vasoconstrict because you want the blood flowing fast. You want to get as much oxygen as quickly as possible. Vasodilating is not going to be happening. Option D says the MR would redirect blood circulating in the brain to organs in the abdominal cavity. This is the exact opposite of the sympathetic nervous system response. This would be something we would see in the parasympathetic nervous response in the rest and digest phase. So blood would be leaving your internal organs and going to your brain, not going from your brain to your internal organs. Question 5 asks, the polymerization of which structural component was analyzed in the experiment described in the passage. And if we refer back to the passage, they mentioned that we are dealing with actin filaments, and that is in paragraph 3. They say that the actin polymerization caused the G-actin to polymerize into the F-actin, and that could be depolymerized by the drug B. And then we look at the answer choices. So immediately I'm thinking microfilaments, but let's see why the other answer choices are incorrect. A states microtubules. Microtubules are dynein and kinesin, and they would not be the actin that we would see within this passage or this study. Option choice C says intermediate filaments, and these are mostly keratin, which is found in the epithelia and in nails and hair. And then option D states thick filaments, when thick filaments refer to the myosin, and although myosin is introduced in this passage, the polymerization that the question is talking about, it says the polymerization of which structural component was analyzed. The myosin weren't polymerized. The actin were actually polymerized. So that's why D may seem correct, but it's actually the wrong answer because they're not referring to the correct thing as stated by the passage. Question 6 asks, what is the likely structure of the amino acid found at position 19 of LC20? So if we go back, that was in paragraph 2, where they said calcium ions bind to the cytosolic protein calmodulin, which activates an enzyme that phosphorylates amino acid residue 19 of the myosin light chain. So whatever amino acid they're talking about has to be able to be phosphorylated. And there's very few amino acids that are phosphorylated. So going through, the ones that we're looking for are serine, threonine, and tyrosine, as well as histidine, arginine, and lysine. And the first three that I said, serine, threonine, and tyrosine, all have an OH group in their R group that could bind to a phosphate that could be phosphorylated. So going through, answer A is actually the structure of glycine. And notice that there is nothing bound to the alpha carbon, no R group besides an H that is assumed to be there. If we look at option B, that is valine. If we look at option C, that is going to be isoleucine. And if we look at option D, that is going to be serine. And notice that there's an OH all the way on the left, and that OH could be phosphorylated. That's why that is the correct answer.